Hello everyone, welcome to 10-Minute Physiology. In today's video, I'm going to try to give you a short introduction to what synaptic transmission is in under 10 minutes. So with that, let's give it a go. So imagine that we have a neuron, and this particular neuron is myelinated. And imagine that we give this neuron a depolarizing stimulus at one of the dendrites. And let's say that this depolarizing stimulus is enough in order to bring the neuron to the threshold potential. Remember that the threshold potential is the cell membrane potential at which the net inward sodium current is greater than the net outward potassium current. And when this occurs, when we get to this threshold potential, an action potential is fired at the node of Ranvier. Now what's going to happen is that this action potential will propagate down the axon until it reaches the synaptic terminal. Now the synaptic terminal is this bulb at the end of the axon where neurotransmitters are going to be stored. Now neurotransmitters are basically chemical signals that are released by an axon from a neuron, which basically transmit a signal from one neuron to another neuron. Now these neurotransmitters can either be depolarizing stimuli or hyperpolarizing stimuli. And we'll talk more about that as we go into individual neurotransmitters in future videos. So the action potential, which has reached the synaptic terminal, will basically cause the release of neurotransmitters. The neurotransmitter will diffuse through the synapse to its target cell and elicit a certain response. Now what I would like to do in this video is give a short overview on what is occurring inside the synaptic terminal. So let's magnify the synaptic terminal in order to see what's going on. So this is the magnification of the synaptic terminal, and what we're going to see is that there are going to be vesicles. Now these vesicles are basically small circular lipid bilayers that are formed inside the cell body of the neuron. So inside the cell body of the neuron, these lipid membranes are created and formed into a vesicle, and they are transported down into the synaptic terminal by microtubules and certain fast transport proteins, which we'll talk about in future videos. So there's going to be two different types of vesicles. The first type of vesicle is going to be a clear vesicle. Now, a clear vesicle is going to be a vesicle that is delivered to the synaptic terminal without any neurotransmitter in it. And when this clear vesicle reaches the synaptic terminal, what's going to happen is that the clear vesicle will be packaged with its neurotransmitter and then stored in the active zone. Now, clear vesicles will tend to contain small neurotransmitters, such as amino acid-derived neurotransmitters. The second type of vesicle is going to be dense granules or dense core granules. Dense core granules will tend to contain proteins, and these dense core granules will tend to be completely synthesized inside the neuron's cell body. So both the vesicles surrounding the neurotransmitter and the neurotransmitters inside are created inside the cell body, and then they are transported down into the synaptic terminal through the microtubule network. Now these types of vesicles will tend to contain peptides, neurotransmitters. So therefore those are the two different types of vesicles, and we'll talk more about that in future videos. So what's going to happen is that this vesicle will move from the end of the axon to a certain area of the synaptic terminal called the active zone. Now the active zones are areas inside the synaptic terminal where a lot of vesicles are stored, and they are basically going to be stored there until they are released. Now inside the axon, we know that there are plenty of voltage-gated sodium channels. So when the action potential reaches the end towards the synaptic terminal, what's going to happen is that the action potential will, of course, cause the opening of voltage-gated sodium channels, which allows sodium to flow in, and the sodium will depolarize the cell. Now, inside the synaptic terminal, we will have a specific type of channel called the voltage-gated calcium channel. Now, the voltage-gated calcium channel in this case will open in response to depolarization, and we'll talk about the specific voltage-gated calcium channel in a future video that is used in the synaptic terminal. So the positive charge will open the voltage-gated calcium channel, which will allow calcium to enter into the cell. This will increase the intracellular calcium level, and this increase in intracellular calcium will basically work on a specific subset of proteins on both the vesicle and the cell membrane of the synaptic terminal to basically interact with each other and cause vesicular fusion. So this increase in calcium will cause the vesicle to fuse with the cell membrane of the synaptic terminal through the calcium interacting with specific proteins, which allows this to occur. And we'll talk about that in another video in the future. So what happens is that the vesicle will fuse with the membrane and then release the neurotransmitters into the synapse. Now, once the neurotransmitter is released into the synapse, it can basically interact with two different classes of receptors. The first receptors are called ligand-gated ion channels, or otherwise known as ionotropic receptors. So basically what's going to happen is that the ligand will bind to the ligand-gated ion channel, so the neurotransmitter, and the ligand-gated ion channel will then open, allowing ions to flow through the cell. Now, these ions can either be cations, or positively charged ions, or anions, or negatively charged ions. And if it's an anion, it will hyperpolarize the cell. If it's a cation, it will depolarize the cell. The second type of receptor that the neurotransmitter can bind to is called a G-protein coupled receptor, or otherwise known as a metabotropic receptor. And we'll talk a lot about these in future videos as we go through specific neurotransmitters. So from this, we see that there are two different types of receptors on the postsynaptic cell. The first are the ionotropic receptors, and ionotropic receptors are ligand-gated ion channels. So in other words, when the neurotransmitter binds to the receptor, it will open the ion channel, allowing certain ions to flow through into the postsynaptic cell. Now, these ionotropic receptors are very quick. They work on a millisecond time scale, and a prime example of this is the nicotinic receptor, which acetylcholine binds to in places such as the neuromuscular junction, or even in the adrenal gland. So therefore, we're going to talk a lot about this nicotinic receptor in another video. The second type of receptor are the metabotropic receptors. And metabotropic receptors are G-protein coupled receptors. Now, because G-protein coupled receptors take a longer time in order to initiate their signal, they tend to work within a second to minute time scale. And a prime example of this are the muscarinic receptors, which also bind to acetylcholine. And there's generally 3 different types of muscarinic receptors, and we'll go through most of them in future videos. So that's all I have for you today for the introduction to synaptic transmission. I hope to see you in the next video, where we'll start delving deeper into synaptic transmission by looking at certain neurotransmitters, looking at how this fusion occurs, looking at specific properties of the different ligand-gated ion channels and metabotropic receptors. So I hope to see you there. Good luck in your studies, and see you next time.